Hello everyone. So today I will start the gametogenesis in plants, and this will end the section B of unit 5, that is Dev biology. And in case of gametogenesis in plants, I'll start with microsporogenesis first, which is the male gamete formation. And so this is a general structure of the stamen, which is the male reproductive unit in plants, and stamen consists of anther and filament. So anther is basically a bilobed structure which is connected by a connective tissue, and the filament is a stalk through which it is connected to the main flower. And this bilobed anther contains two pollen sacs each, which pollen sacs are known as microsporangia. And so here you can see that they are tetrasporangiate, 4 sporangia. And in case of a transverse section in anther, as you can see that the sporogenous tissue lies here in the middle part, and the two lobes are connected by a connective tissue. Inside the sporogenous tissue, the sporocytes are formed, and that is covered by a number of layers, protective layers, the epidermis, the endothecium, and the tapetum, middle layer, and the tapetum. So tapetum is basically providing nutrition to the sporogenous tissue. So inside the sporogenous tissue, there are now some cells which get differentiated into microsporocytes, and these microsporocytes, they undergo mitotic division and form a number of 2n mother cells, like microspore mother cells. These microspore mother cells, they have a layer called the callus wall that breaks the internal connections among these cells. Now each microspores, the mother cell, can produce 4 haploid microspores by means of meiosis. So these microspores are arranged in a tetrad-like formation, and microspore is actually the pollen grain that we commonly call as pollen grain, and this process of formation of microspores is known as microsporogenesis, and this is the first cell of the male gametophyte. And here if you see that this is a released microspore, it has two outer coverings, one which is discontinuous and thick, that is known as exine, and the one which is thinner and continuous, that inner one is known as intine. So this microspore, after undergoing mitosis, it forms two cells, the vegetative cell, larger one, and the generative cell, smaller one. Next, the generative cell usually detaches itself from the outer covering and starts freely moving in the vegetative cell. This stage is known as a bicellular pollen, and then it undergoes a second mitosis, and this becomes a tricellular pollen because the generative cell divides into 2 sperm cells. So next, if we go into much more detailed structure, the sporoderm consists of the outer exine and the inner intine, like I already said. And the gaps where the exine is missing, those gaps are known as germ pore. That is helpful when the pollen tube is coming out. So this I have already mentioned, that the larger one is the vegetative cell and the smaller one is the generative cell. The generative cell starts dividing and becomes 2 male gametes. So this pollen actually gives rise to a pollen tube when it reaches the stigma of the flower. Stigma is the part of female reproductive or unit. So as you can see that the tube cells grow into the pollen tube on successful germination, while the generative cell goes into the ovary through the pollen tube. So this is what happens. This is the female reproductive unit, or the carpel, as we can say. The pollen, when it sits here on the stigma, it forms a pollen tube, and the generative nucleus and the tube nucleus is coming through it, and this tube is entering into the ovary of the female reproductive unit, and the 2 sperm nuclei are also getting released over here. So when I do the female gametogenesis, then it will be much more easier to understand. So in this way, the male gametophyte is developed. Next, moving on to the female gametogenesis or megasporogenesis in plants, that is, this is a pistil, and it is the female reproductive unit in a plant which consists of the stigma, the style, and the ovary. So the ovary contains ovules, and each unit of the ovary is known as a carpel, and if there are a number of carpels together, it is known as a pistil. So in case of an ovary, if you can see that according to the position of the micropyle and the chalaza, that is, the micropyle is this portion and the chalaza is this portion. So according to the position of these two poles, the ovules can be classified into the following types. So here the chalaza is basically the part where the ovule or the ovary is connected with the filament, the style, and this part is the where the integuments are open, micropyle. This portion is useful when the pollen tube wants to enter the ovary. So here if you can see that there are a number of cells which form the embryo sac in case of females. The first one is the antipodal cells, 3 over here, then the definitive nucleus here, and here 2 synergids and 1 egg cell is present. So hilum is the connection over here, and the funiculus is the attachment of this entire structure with the style. So according to that, the ovary or the ovules are classified into the following types.